So, hello everyone. This is the term 1 week 6 periodic college board quiz. Okay, so this is most probably going to be what most of our exam is going to be about. It's not that hard صراحة. It has some past AMS questions, some new questions. You just have to open up your brains and think, because AP style questions require a lot of critical thinking and require proper understanding of the actual concept. So make sure you understand the concepts before doing this quiz. Okay, so the CQD video, you should have did it. You should have did the past AMSs. You should understand what's going on before you go into this quiz. Okay, so let's start. So researchers claimed that a particular organelle originated from a free-living prokaryotic cell that was engulfed by a larger cell. Figure 1, a model showing a cell engulfing a smaller cell. Which of the following provide best evidence to the researcher's claim? So the organelle has a double membrane. Obviously, if the organelle has a double membrane, it was eaten by another atom, another organism. Okay, so the CFTR protein is made of 1480 amino acids linked together in a chain. Some humans produce a version of the CFTR protein in which phenylalanine, an amino acid, has been deleted from position 508. So there's this big chain of 1480 amino acids, and then there's a mutated amino acid, like there's a mutated protein. So this protein CFTR is the normal one, it has 1480 amino acids. There's a mutated protein, a mutated version of CFTR that has only 1479 amino acids. Obviously, this will have a very significant change. Okay, so it will affect the primary structure, the secondary structure, the tertiary structure. It will affect all structures of the protein. Okay, so it will affect the primary, secondary, and tertiary structure of the CFTR protein. Okay. So which of the following describes the key difference among the 20 amino acids that are used to make proteins? So in amino acids, okay, there are 10 amino acids that are hydrophobic and 10 amino acids that are hydrophilic. 10 amino acids that hate water, 10 amino acids that like water. That's the difference between them. Some are hydrophobic, and that is the correct answer. Okay, 3 amino acids here. So this shows you 2 different types of proteins. One of them is the normal one. It has the cysteine in the middle, and then one of them is mutated. It has valine in the middle. Obviously, the R group of the new amino acid, valine, has different chemical properties than the R group of cysteine. Obviously, as you can see, they're not the exact same thing. See how cysteine is and see how valine is. They're 2 different things. Okay, and this will cause the protein to misfold and not function properly in the cell. So just like we said, when we remove the R group, when we change the R group, things get messed up and the protein can't function properly anymore. Okay, so this is the correct answer. The carbohydrates, glucose, galactose, and fructose have the same chemical formula. Okay, but even if they have the same chemical formula, see here, they have different structural formulas. They don't all look the same. If you look closely, it's not all the same. Okay, there are some subtle differences, but these subtle differences make a big difference about the properties. Okay, so they have different arrangement. So since they have different arrangements of carbon, hydrogen, and oxygen, they have different properties. Oh, and by the way, guys, also number 1, number 2, and number 56 are FRQ, free response questions. It's basically written. I'll do them at the end just because it's better to finish the multiple choice first. Okay, which of the following best describes how the carbohydrate polymer is being built in figure 1? Okay, so obviously monomer number 1, monomer number 2 are being put together by a by a dehydration reaction. Okay, dehydration synthesis, condensation, and a water molecule is removed and the covalent bond is formed. So that is correct. Here, which of the following statements best describe how organisms such as rabbits obtain carbon from necessary for building biological molecules? So as you can see here, you have grass. Okay, the grass has carbon. The rabbit eats the grass. the rabbit will then have carbon. So the rabbits eat plants and break down plant molecules to obtain carbon and other atoms that they rearrange into new carbon-containing molecules. So that is the correct answer, obviously. And what's it called? So the plant originally has carbon. The rabbit eats the carbon. It breaks down the plant, and then it extracts the carbon. Okay. So plant has carbon. Rabbit does not have carbon. Rabbit goes, eats the plant, breaks down the plant, extracts the carbon and the other nutrients it wants, and then it rearranges it into its own molecules, into like new carbon-containing molecules. Okay, which of the following best describes the relationship between the size and shape of a cell and the ability of the cell to move substances across the cell membrane? Okay, so if the cell is small, it can move very easily. And if it has an irregular shape, meaning that the shape can be shifted, not a regular shape. A regular shape means that the cell has this shape, it cannot change. So basically, if I had a plant cell, see, a plant cell, it's like this. It's a box because of the cell wall. It's rigid. It can't move. It can't change. But if I had this squishy thing and it can change, it can become like this, it can become like that, and it's small, it means that it can move easily and it can change the shape very easily. So it's very easy to like move around. Okay? Correct. So which of the following would most likely occur if cattle lost the ability to maintain a colony of microorganisms in their digestive tract? Whenever you're doing any exam, really, it's always better to read the question first and understand what they want before going and reading the given. Okay, I like to do that, especially if you're doing your SATs, okay, and you're going into English, especially the English section. Always read the question first and then look at the given, okay? So they want, so what would happen if we lost the microorganisms? If the cattle lost the microorganisms, sorry. So let's read the given. Different polysaccharides are used by plants for energy storage and structural support. The molecular structures for two common polysaccharides are shown in figure 1. Starch is used by plants for energy storage, and cellulose provides structural support for cell walls. The monomer used to construct both molecules is glucose. Okay, so we have starch, we have cellulose. Both the primary building blocks are glucose. Okay, but as you know, cellulose is beta glucose and starch has alpha glucose. Yeah, and cellulose, obviously each glucose is like flipped over. So as you can see here, see, look at the position of that. It became down, then it went up, then it became down. So it's flipping the thing. Okay. So a study determined the effect of two different digestive enzymes, A and B, on these two polysaccharides. Okay. So in the test tube 1, we had cellulose. We added enzyme A, and then there was no more glucose left, which means it's bad because you'll still need glucose. So when enzyme A was added to cellulose, there was no more glucose. When enzyme B was added to cellulose, there was still glucose, which is good. When enzyme A was added to starch, we still had glucose. Good. When enzyme B was added to starch, we didn't have any more glucose, which is bad. Mammals do not produce digestive enzyme B. Okay, so us as mammals, we don't produce enzyme B. But sheep and cattle, these are the two types of mammals that they contain microorganisms in their digestive tract that produce enzyme B for them. Okay, so in cattle, they have microorganisms in the digestive tract that create the enzyme B for them. Okay, so what would happen if the cattle lost the microorganisms in their digestive tract? Well, cattle would no longer be able to use cellulose as a primary source of glucose. Why? Because if we don't have enzyme B, okay, if we don't have enzyme B, that means that cellulose would just stay cellulose. So we couldn't use cellulose as a storage point because B doesn't attach to cellulose to break it away and give us the usable glucose, because we can't use cellulose as a source of energy. We can only use glucose. To obtain the glucose, we need an enzyme to go attach itself to cellulose or starch, break it down into glucose for us to actually use. If we don't have if the cattle doesn't have the microorganisms, it can't produce enzyme B, which means that it can't use cellulose as a primary source of glucose. Okay, that is the correct answer. Which of the following best describes the process that adds a monosaccharide to an existing polysaccharide? Okay, so obviously a specific enzyme. So this is basically just telling us to describe dehydration reaction. So a specific enzyme removes the H, then the other enzyme removes OH, then they create a bond, and you get water as a byproduct. That is the correct answer. By the way, guys, sorry about my voice. I don't know what's happening with me and my throat. I'm getting crazy voice cracks, and it's just messed up. So sorry about that. So based on figure 1, which of the following best compares the atomic structures of starch and cellulose? So starch and cellulose are composed of repeating glucose monomers. However, in cellulose, every other glucose monomer is rotated 180 degrees. Obviously, as you can see here, just like I pointed out at the start of the question, this is linear, so it's straight. So see how it's up, 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 up. So it's one chain of the same exact thing, same position, okay? But here in cellulose, one is up, one is down, one is up, one is down. So it's rotated, okay? It's rotating, ماشي? So that's why we choose B. Next. So researchers have proposed a model of chloroplast evolution. According to the model, chloroplast evolved from a small prokaryotic organism that was engulfed by an ancestral eukaryote. So remember how chloroplast evolved? So we had a eukaryote that went and ate an autotrophic prokaryote, okay, to get chloroplast. What best supports the model? Chloroplasts and some prokaryotes share similar photosynthetic reactions. So if the origin of chloroplast was the autotrophic prokaryote, then if the chloroplast of eukaryotes and the autotrophic prokaryotes undergo similar photosynthetic reactions, that means that it was actually engulfed. So researchers compared similar proteins from related organisms in different habitats. They found that the proteins from organisms living in harsh environments had a greater number of cysteine amino acids than did proteins from organisms not living in harsh environments. So obviously you'd have disulfide bonds in cysteine amino acids. So that's the only type of amino acid that would have disulfide bonds, the sulfide bonds. Okay, those bonds are very stable, which makes them very good for harsh environments, okay, where the temperatures are high and all of that. So you'd have a very good stable base, which is good for the protein. Okay, so the change leads to increased protein stability. Remember, disulfide has everything to do with stability. Sorry guys, but my throat is clogged. I don't know what's going on. So the change leads to increased protein stability because of an increased number of SS bonds in the tertiary structure of proteins. Okay, and the answer is correct. So which of the following statements best describes the molecular changes that occur during the reaction? Okay, so here, as you can see, what's it called? As you can see, wait, okay, 1 second, what's going on? Okay, so which of the following statements best describe the molecular changes that occur during the reaction shown in figure 1? So obviously, a hydroxyl group from one glucose molecule and a hydrogen atom from the other glucose molecule are removed and one water molecule is formed. So you see here, water molecule formed. Obviously, you'd have to remove a hydrogen atom and an OH atom to get the bond. So it's a dehydration reaction. Dehydration synthesis. Okay. And the answer is correct. Next one. Which of the following is a common feature of the illustrated reactions showing the linking of monomers to form macromolecules? Which of the following is a common feature of the illustrated reactions showing the linking of monomers to form macromolecules? So monomers are joined by a covalent bond and a water molecule is produced. So obviously, when you want to join two molecules together, you will have to release water, remove the OH and then H, and make a bond, a covalent bond, okay, a covalent peptide bond to form the amino acid, and it is called the dehydration process, and it is correct. Which of the following best describes a cell that is likely to have an extensive Golgi complex? So, extensive Golgi complex, So what the Golgi complex does, it packages and modifies the protein that was created by the ribosomes. So the ribosomes or the ribosomes on the endoplasmic reticulum. I am a horrible artist, horrible. Okay, so you have ribosomes on the RER, the rough endoplasmic reticulum, and you have ribosomes just in the cytoplasm. Okay, they create proteins. These go to the Golgi complex, pancakes. I don't know how to draw. This is the Golgi complex. It doesn't actually look like this, but yeah. Okay, it goes to the Golgi complex here, and then the Golgi complex modifies the proteins, packages, and sends. Okay, which is why a stomach cell that produces and releases digestive enzymes would have an extensive Golgi complex. Why? Why enzymes? Because enzymes are proteins. Okay, remember that. And the answer is correct. So, which of the following best describes the structural similarity between the two molecules shown in figure 1 that is relevant to their function? Okay, so both molecules contain nucleotides that form base pairs with other nucleotides, which allow each molecule to act as a template in the synthesis of other nucleic molecules. Okay, guys, everything in this sentence is correct. Okay, everything in that sentence is correct, obviously. Okay, let's go through each sentence and say what's wrong. So both molecules are composed of the same 4 nucleotides? No, because in DNA we have adenine, guanine, thymine, cytosine, but in the RNA we have, instead of thymine, uracil. Okay, both molecules are composed of the same 5-carbon sugar, which allows each molecule to act as a building block for the production of polysaccharides. What do you mean production of polysaccharides? It doesn't produce polysaccharides. Same here, same thing here. They say the synthesis of proteins and lipids. That's wrong. Okay, so this is the only one that's correct 100%. Which of the following statements is correct about the molecule shown in figure 1? Okay, so from here you can obviously see that cytosine, guanine, adenine, thymine. You see thymine, that means it's DNA. Okay, and it's DNA because of the nucleotides present. Okay, so we just checked what nucleotides are present. When we saw thymine, it is definitely DNA, and it is correct, thank God. Figure 1 represents a nucleic acid fragment that is made up of 4 nucleotides linked together in a chain. Okay, so here you have 4 nucleotides in a chain. Which of the following characteristics of figure 1 best shows that the fragment is RNA and not DNA? So the identity of each nitrogenous base. Okay, correct, RNA contains uracil, whereas DNA contains thymine, just like I said. I know that you can't actually see the uracil or the thymine or whatever. I know that they didn't label it for you. But if you think about it, if you look at the other options here, the 5' to the 3' orientation, they both have 5' to 3' orientation, okay? The charges on the phosphate groups, the RNA and DNA both have similar charges of phosphate groups, okay? And the type of bond linking the nucleotides together is the same. They're both the phosphodiester bond, okay? Which is why the identity of each nitrogenous base is the one that makes the most sense in this case. Which of the following statements best supports the claim that certain organelles within eukaryotic cells evolved from free living prokaryotic cells? Obviously, some organelles contain their own DNA that is more similar to prokaryotic DNA in structure and function than to eukaryotic DNA found in cell's nucleus. Okay, so just like you know, I'm not gonna draw the mitochondria and chloroplast because I'll just embarrass myself, صراحة. So what's it called? As you know, that both the chloroplast and the mitochondria have their own 70S ribosomes and their own circular DNA. What does that resemble? That resembles the prokaryotic cells, which is why they say that we have common ancestry, blah blah blah, you know, things, 8 other things to make, you know. This was a previous AMS question, okay? So which one would be solid at room temperature? They're basically asking you which one is saturated. Remember, saturated only have single covalent bonds, not double covalent bonds. This is unsaturated, this is saturated. Okay, so as you can see here, there is only single covalent bonds. Don't tell me that this is a double bond. No, this is a double bond between C and O, not C and C. We only look at the carbons, we don't look at O. So we only care about the carbons, we don't care about this. This is with O, نحن ما خصنا. We care about these, okay? If it has single bonds with the C, then it's saturated. If it has double bonds with the C, it's not saturated. Unsaturated, okay? It's obviously the palmitic acid because the absence of the carbon double bonds allows the molecules to pack closely together. So if you don't have double bonds, you will have closer packing, which makes it solid at room temperature. Which of the following statements describes a feature shared by mitochondria and the endoplasmic reticulum that increases the efficiency of their base functions? They have highly folded membranes. Guys, inside of the mitochondria, you have these highly folded membranes. They're called cristae, okay? And they increase the surface area of the mitochondria, makes it more efficient. Same thing in the endoplasmic reticulum. It looks like this. It's like a weird highway, okay? It's folded, so it increases the, efficiency and surface area, okay? Based on the information provided, Gaucher disease results most directly from a defect in the function of which of the following organelles? So let's see what Gaucher disease is. Gaucher disease is an inherited disorder in which cells of the body are unable to break down. There we go. Unable to break down a particular type of lipid. Now, I know when you see lipid, you're going to go smooth endoplasmic reticulum. That's wrong. Because the smooth endoplasmic reticulum synthesizes lipids, and it detoxifies. Okay? Detoxifies. It has nothing to do with breaking down particular types of lipids or anything else, really. But the lysosome, the cleaners of the cell, break down, which is why the lysosome is responsible for this particular disease, and which is why it is the correct answer. All eukaryotic cells contain at least one Golgi complex, typically located in the cytoplasm and near the endoplasmic reticulum. So which of the following best describes a process that occurs within the Golgi complex? Enzymatic modification of newly synthesized integral membrane proteins. So basically, all they're telling you here, it's that it takes the proteins, modifies them. They are newly synthesized proteins. So newly synthesized proteins undergo modification. And yeah, that's it. That's all they're telling you there. So newly synthesized proteins undergo modification. That is the function of the Golgi complex, and that is it. That is why it is the correct answer. Okay, here some people already asked me about, oh no, that was number 28. This is pretty straightforward. Guys, all you have to do basically when they ask you about the effectiveness or the efficiency, okay, you have to divide surface area by total volume. So you have to do division, okay? You have to divide these two together. Okay? So, and it's always surface area over volume when you're trying to find the efficiency, okay? So you do 40 over 20, 60 over 10, 80 over 30, 100 over 20. The biggest answer is the most efficient. The smallest answer is always the less efficient, okay? 60 over 10 gives you the biggest answer, which is why it is cell 2. Okay, so here, listen. This HS, it's like a disorder in red blood cells, and it makes the diameter a bit smaller, okay? So this is the normal diameter, this is the smaller diameter, okay? Here they already gave you the information to find the efficiency of a normal red blood cell, okay? What they're asking you is which of the following provides an accurate calculation of the surface area to volume ratio of an HS red blood cell. And it tells you, after you find the ratio of the HS blood cell, the ratio being surface area over volume, tell us which one is more efficient. Is it the normal red blood cell or the HS blood cell? The normal red blood cell, they already gave you the information here. They already gave you the surface area and they already gave you the volume. Surface area, volume. But here they gave you the diameter. What do I do with the diameter? I'll tell you what to do. To find the surface area, let's take into account that a cell is a sphere, okay? To find the surface area of a sphere, it is 4 pi r squared. You plug in the 6.7, but you don't plug in the 6.7. You divide it by 2. Why? Because it's r squared, not d. This is the diameter. The r is 6.7 over 2. So r is 6.7 over 2. Diameter is just 6.7, okay? So you plug in, you find the answer, which will be surface area. Then volume is 4 over 3 pi r cubed. Volume, okay? The answer is volume. Okay, so now you have surface area and volume. When you divide these two together, surface area and volume, you will get 0.89, which is the only correct number. And the normal one is 136, what's it called? 136 over 91. And if you plug in 136 over 91 into your calculator, you will get that it is 1.49. And which one is bigger? 149 is bigger. Sorry, 1.49, 149. 1.49 is bigger, meaning that the normal blood cell is more efficient. Okay? So that is the correct answer. Oh, and by the way, you keep it in micrometers. Why do you keep it in micrometers here? Because look at the unit. It's in micrometers, so you can just keep it in micrometers. Okay. Water molecules are polar covalent molecules. There is a partial negative charge, blah, 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 blah. Okay, which of the following best demonstrates the arrangement of hydrogen bonds? Guys, I have done this so many times. It obviously isn't this is just, no, it's between a hydrogen and an oxygen, not a hydrogen and a hydrogen. This is fine, but that's not a hydrogen bond. Okay, this is diabolical. This is an abomination. That, I don't know what that is. It's two O's together. That does not happen. Same thing here. That does not happen. It has to be like that. Remember, just like how I did it in the CQD video, the O is connected to the H because the O is slightly negative, the H is slightly positive, and they can't be in the same water molecule. When they are in the same water molecule, it's called a polar covalent bond. Covalent bond. When it is between two different water molecules, it's called hydrogen bond. Okay. Correct. Water and ammonia interact to form hydrogen bonds as represented in the figure. So why is the hydrogen bond formed here? Guys, by the way, hydrogen bonds are not formed only between H and O. It has to have an H, obviously, because it's hydrogen bond, but it can bond to any slightly electronegative, oh my god, I don't know how to draw the symbol as well. Slightly electronegative atom. Anything can be inside here, anything. Okay, as long as it is slightly electronegative, just like here, which is why the hydrogen bond is formed. So the nitrogen has a partial negative charge, hydrogen is partial positive, they attract, bang, you have a hydrogen bond. Oh, and by the way, for any hydrogen bond, they can't be in the same molecule, they have to be in different molecules. If they're in the same molecule, then it's a polar covalent bond. Okay, just like in the case of water. Which of the following best explains why hydrolytic enzymes are typically transported to the lysosome after they are synthesized by ribosomes? Okay, now ask yourself, what do lysosomes do? They break things down, right? So what are the enzymes that are taken to the lysosomes supposed to do? Break down things, right? So these enzymes are essential for breaking down organic molecules, which is correct. As shown in the diagram, when environmental, guys, this is an AMS question. They're asking you what the function of ice is. So the water molecules in ice are farther apart because ice is less dense than water. So the ice floats. It maintains the water temperature that's down and keeps the cold out. Okay. Which feature of Model 1 best illustrates how biological information is coded in a DNA molecule? Okay, so as you can see here in diagram 1, Model 1, it shows you the base pairings, which is why it's the linear sequence of base pairs, which is the correct answer. Changing the shape of morphology of the mitochondrial inner membrane can change the efficiency of mitochondrial function. Okay, so which of the following outcomes will most likely result from a change in the shape of mitochondrial inner membrane from a highly folded surface to to a smooth, flat surface. So guys, remember I told you that the inner membrane of the mitochondria looks like this. Okay, if it becomes like this, which has more surface area, this or that? This has a higher surface area, more efficient. This has less surface area, less efficient. Okay, so mitochondria will be less efficient if it becomes flat because of the less surface area in the inner mitochondrial membranes. Correct. Allah, I have to read the question. The figure shows a process by which a cell might absorb food from its surrounding environment and break it down for use as a source of energy and matter. The process involves lysosomes, which are membrane-bound organelles that contain hydrolytic enzymes. Activation of the hydrolytic enzymes requires an acidic pH, and lysosomes maintain an internal acidic pH by using ion pumps. Basically, guys, these ion pumps maintain the acidity of the lysosomes. And why do lysosomes have to be acidic? So they can activate their hydrolytic enzymes, so they can actually break down things. So they can actually break things down. Okay? What happens when you lose the ion pump? The internal pH will increase, remember, because acidity decreases, alkalinity increases. 7.1, this is acidic, very acidic, this is very alkaline. No. 0, I think, yeah. This is very acidic, this is very alkaline, this is neutral. Okay? So if it increases, it becomes less acidic, which means that's bad, which means that the activation of the hydrolytic enzymes will not be achieved, and it will interfere with the digestion of food. Okay, some viral infections can lead to the rupture of the lysosome membrane. Which prediction of the effect of the disruption of cellular compartmentalization is most likely correct? So if the lysosome loses its membrane, that means everything inside the lysosome will be exposed to the rest of the cell. And just like I said in the CQT video, it's very important that we compartmentalize, especially the lysosome, because if that material inside the lysosome is exposed to the rest of the cell, it means that the hydrolytic enzymes will be released. And when the hydrolytic enzymes are released, it will digest your cell, which will cause the cell to die. Correct. Which of the following claims is scientifically accurate and consistent with an observation that a decrease in lysosome production within a cell leads to a decline in mitochondrial activity? So, fewer lysosomes will be available to break down macromolecules to provide the necessary nutrients for cellular respiration. So, to get the necessary nutrients for the cellular respiration that happens in the mitochondria, you need lysosomes to break down the macromolecules to actually get those nutrients. If you don't have lysosomes, you will not get that nutrients, which will decrease the mitochondrial activity. Which of the following observations best supports the model? So let's read the model. The endosymbiont theory proposes a model for the evolution of mitochondria. According to the model, an ancestral eukaryote engulfed a small free-living prokaryote. The engulfed prokaryote then formed an endosymbiotic relationship with the eukaryotic host. So, mitochondria and some prokaryotes share similar metabolic reactions that produce ATP. So, just like in the case of chloroplasts, if the mitochondria and the prokaryotes have similar metabolic reactions, that means that the prokaryotes and the eukaryotes have the same ancestors, like it came from the same source. So the mitochondria came from the same source, which was a prokaryote. Okay? And yeah, that's it. So next. Which of the following observations best supports the claim that mitochondria evolved from once free-living prokaryotic cells by the process of endocytosis? So, mitochondria are surrounded by a double membrane. If you have a double membrane, two membranes, where did the second membrane come from? Where did that come from? Obviously, something engulfed the cytoplasm. So something came in here, engulfed it, وما بعرف شو, and it gave it the second membrane. Okay? So, surrounded by a double membrane. Okay. Phosphorus is an important nutrient for plant growth. Figure 1 shows Arabiposis thaliana, plants grown under phosphorus sufficient left. So the phosphorus sufficient is on the left, the phosphorus starved is on the right. Okay. So basically what they're telling us is here there is no phosphorus, here there is phosphorus. Oh, sorry, relax. Sorry. Here there is no phosphorus, here there is phosphorus. Sorry, guys. So remember, left has phosphorus, right does not have phosphorus. So, which of the following is the most likely reason for the difference in leaf growth? Okay, so the phosphorus-starved leaves was unable to synthesize both the required nucleic acids and lipids, limiting growth. So if we remember, phosphorus is one of the building blocks of nucleic acids and phospholipids, meaning that if we don't have phosphorus, we won't be able to have nucleic acids or phospholipids. That will limit our growth. That is why the plant on the right is smaller than the plant on the left. The plant on the left has phosphorus, so it has nucleic acid, it has phospholipids. It can grow way more efficiently than the one on the right. Correct. Which of the following best describes the formation of the bond? Here, obviously, enzyme attaches, OH is gone, H is gone, H2O is produced, peptide bond is produced. Why is it a peptide bond? Here you have the amino terminal, here you have the carboxyl terminal. When these two terminals bond, it's going to be a peptide bond. So covalent bond is formed between a carbon atom and the nitrogen atom along with the formation of H2O. Okay, see here, the bond is formed between the carbon and the nitrogen. Here, OH is gone and H is gone, so it becomes H2O. H2O is released. Peptide bonds are also covalent bonds. They are a type of covalent bond. Okay, so which of the following is accurate description of the process shown in figure 1? Isn't this the exact same thing that I've done? Yeah, it's the exact same thing. It's called the dehydration reaction, guys. Okay, so students conducted a controlled experiment to investigate whether sawdust... Wait, let me read the question first. The observed differences between the groups most likely resulted from differences in the ability of seedlings to produce which of the following monomers? Okay, so maybe we don't have to read the question here. Look here. Seedlings in potting soil and seedlings in sawdust. Here, look at that. The seedling height in potting soil is 18.5, but the seedling height in sawdust is 4.8. That means sawdust is missing something that soil has. So soil has something that sawdust doesn't. Okay, and as you can see here, the leaves have a dark green color and are normal in size, but the leaves have a grayish color and are small in size in sawdust. So sawdust is definitely missing something that soil has. Let's go down and see. Is it missing, what's it called? Ya Allah. Oh my God, I forgot what I was gonna say. Oh, okay. So it's obviously missing proteins and what's it called? And nucleic acids. Because proteins build up the cell a lot. Proteins are responsible for building up your cells. So are nucleic acids, which is why it will be that. Okay? And also, if you read more, students conducted a controlled experiment to investigate whether sawdust provides enough nutrients to support plant growth. The students separated 10 nearly identical sunflower seeds into two groups. They grew the seedlings in the first group potting soil and the seedlings in the second group in sawdust, composed mostly of cellulose. After 20 days, the students recorded observations. Oh, okay, so they don't tell you that one of them has phosphorus and one of them doesn't. Okay, guys, basically, what's it called? Sawdust will not provide the sufficient amounts of nitrogen and phosphorus, resulting in the seedlings having a reduced ability to provide both the monomers shown, amino acids and nucleotides. You'll know that it's amino acids and nucleotides because of the growth and because of the dark green color. Okay? So just keep that in mind. Tamam? When you see growth, it's most probably proteins and nucleic acids. Okay? And phospholipids sometimes as well, but they didn't give you an option for phospholipids here. Okay? Okay, so which of the following best depicts the tertiary structure of the two proteins in water? Okay, so guys, here all they're telling you is you have two different proteins. Okay, you have, this is the original protein with these hydrophobic amino acids and the rest is hydrophilic. Okay, in the second one they removed the hydrophobic ones, they removed them, they're gone here, they just kept these. Okay, they're asking you what the tertiary structure will be. Remember the tertiary structure with the, what's it called, with the hydrophobic interactions? The hydrophobic ones are always on the inside. They're shy, they clump together on the inside, while the very social non-polar hydrophilic things are on the outside. Okay, so it's the exact same thing, like they look very similar, so it will be this one. Obviously it won't be this, it never protrudes like this, it doesn't do that, and it doesn't do this either. Like, that's not tertiary structure, it has to be coiled up, and that's just a circle. I don't know what that is. Muscle cells have high ATP demands. Which of the following is a scientific claim about how the structure of mitochondria in muscle cells could be different than it is in other cells because of the high energy demands of mitochondria? So, the inner membrane of mitochondria in muscles should have more folds to increase the surface area, allowing more ATP to be synthesized. Now, there is another one that says the outer membrane of the mitochondria of muscle cells should have more folds, increasing the surface area for faster diffusion of molecules from the cytoplasm. Guys, if you have faster diffusion of molecules from the cytoplasm, fine, amazing, okay. But in the end of the day, if you're a muscle cell, you need more energy, you need more ATP. It doesn't have anything to do with the diffusion. So increasing the folds in the outer membrane won't solve your problem. You have to increase the folds in your inner membrane to actually increase the production of ATP. Oh my god. Ya Allah. Okay, let's read the question. Let's read what they want first. Based on figure 1, which of the following statements best explains why the nuclei sample had a higher level of radioactivity than any of the rinse water samples? So they're comparing rinse water and nuclei, and they're comparing their radioactivity. So let's see what they did. A researcher conducted an experiment in a brightly lit aquarium containing a large population of single-celled green algae. Water containing radioactive phosphorus was added to the aquarium. Okay. After 7 days, the researcher took a water sample containing the algae and separated the algae from the radioactive water. Okay. The researcher added deionized water to rinse the algae and saved the rinsed water, and then repeated the process twice. The rinse water from each of the 3 rinses was saved as rinse water 1, rinse water 2, and rinse water 3. The researcher also removed and kept the nuclei of the algal cells. The nuclei were mixed with deionized water, and a drop of the mixture was placed on a piece of filter paper. A drop of radioactive water and a drop of each of the rinse water samples were also placed on the same paper. These samples were exposed to X-ray to determine the radioactivity. Okay, so the darker the color, the larger the size, the higher level of radioactivity. Okay, basically, radioactive water contained phosphorus. So, and as you can see here, the radioactive water, the normal radioactive water that wasn't rinsed, that didn't undergo anything, the pure radioactive water is very dark. So it has a lot of phosphorus. What does our nucleus also contain? It contains DNA and RNA. DNA and RNA are nucleotides. What do nucleotides contain? Phosphorus. Which is why the nucleus is more radioactive than the rinse water 1, rinse water 2, and rinse water 3. Way more radioactive. Okay? So phosphorus is used in building of nucleic acids in the nucleus, which is why it is more radioactive, because phosphorus is what makes things radioactive in this case. Which of the following statements best describes the formation of complex carbohydrates? Monosaccharides are joined together through covalent bonds. That's fairly straightforward. The monosaccharides join, you get a polymer, which are the polysaccharides. Which of the following best describes the structures of carbohydrates? They occur as monomers, chains of monomers, and branched structures. So carbohydrates, you have monomers, polymers, and branched structures, because the polymers are also branched. So yeah, like amylopectin is branched, so that's why. Which statement is the most accurate description of the reaction in figure 1? Okay, so as you can see here, you have water being added. Look, water is being added. It's not being removed, it's being added, okay? So when you add water, you're usually breaking things down. Well, you're always breaking things down, okay? So you have hydrolysis, you have the two amino acids being broken apart through hydrolysis. So it represents a polypeptide chain that is broken down through a hydrolysis reaction. Correct. Which of the following statements best describes the difference between proteins and nucleic acids? So proteins contain sulfur while nucleic acids do not. Okay. Correct. Okay, so which of the following conclusions about the radio-labeled amino acid is best supported by the results of the experiment? Okay, so let's read what the experiment is. An experiment researchers provided a radio-labeled amino acid to living plant cells. So they got a labeled amino acid and added it to plant cells. After 1 hour, the researchers determined the amount of the radio-labeled amino acid that was in each of the several subcellular compartments. The results of the experiment are reported in the table. So this much was found in the nucleus, this much was found in the mitochondria, this much was found in the ER, and this much in the cytosol. The mitochondria had the biggest concentration, meaning most of it was used in the mitochondria, meaning it was used to manage metabolic reactions. Okay, it was mostly incorporated into proteins that regulate and manage metabolic reactions. Correct. A researcher is studying a specific type of cellular defect in animal cells. She notices that cells with this defect have reduced energy storage and decreased cell membrane stability. Which of the following predicts the type of cellular defect that is the most likely cause of the researcher's observation? So, a defect in fatty acid synthesis reducing the formation of fats and phospholipids. Okay. Why? Because she notices that the cells have reduced energy storage and decreased cell membrane stability, and the thing that gives the cell membrane stability is phospholipids, and reduced energy storage is also due to fats, which is how we store our long-term energy, through fats, and phospholipids is the thing that gives our cell membrane its stability. Okay, which is why that there was a defect in those two. Which cellular component will be found in the widest range of organisms in the sample? So they're comparing a prokaryote and a eukaryote, and we know that both eukaryotes and prokaryotes have ribosomes, which is why this one is correct. A certain type of specialized cell contains an unusually large amount of rough endoplasmic reticulum. So which of the following functions is the cell type most likely specialized to perform? So what does the RER do? It produces and secretes proteins. What is the cell most likely to do? Produce and secrete proteins. Which statement correctly indicates the cell that is able to more efficiently exchange materials with the external environment and provides a correct explanation? Okay, so guys, here, listen, they're asking us for efficiency. Okay, they're asking us for efficiency. So what we're going to do is we're going to find our ratio, which is surface area over volume. Now, they only gave us the radius. They were nice and they gave us the radius. They didn't give us the diameter, so that's better. They gave us the radius, tamam? Okay. Now our job is to find the ratio, which is surface area over volume. So surface area over volume. This is our ratio. Okay. Now, here 4 pi r squared for the bacterial cell is 3, so 4 pi 3 squared, you get the surface area. 4 over 3 pi r cubed, 4 over 3 pi 3 cubed, you get volume. You do surface area over volume and you get the ratio. Okay, same thing here, 4 pi r squared, 4 pi 100 squared for the human egg cell, you get surface area. Then same thing for the volume, 4 over 3 pi r cubed, 4 over 3 pi 100 cubed, you get volume. You do surface area over volume, you get the ratio. You compare the ratios, you see the bigger one, which will be the bacterial cell, actually by 33 times. So the bacterial cell is 33 times more efficient than the human egg cell. And the reason why is because this will be 33 times bigger than that. So, and the bigger one is always more efficient. So yeah, the bacterial cell, because it has the largest surface to volume ratio. Okay. Next. Okay, so now we are in the point where we are going to start doing the FRQs. Guys, just a little bit about the FRQs. especially if you are going to be doing the external exam next year, the APs. I have done 2 APs, okay, alhamdulillah, and the most important thing in APs are the FRQs. The thing that always saves me is the FRQ. You have to know how to write. You can't go in, write 3 words, and expect to get the mark. Even if you covered the points that they want, if it's not written in proper English, they won't give you the mark. They won't even grade it. They will literally not grade it for you. You have to have it in a sentence that makes sense. Proper grammar, proper English. So start practicing how to use the fancy words, how to write it in nice big sentences, and to make everything clear and defined. Every single boring detail has to be in here. Okay? So here they're not really asking about a lot, and I didn't do it properly because I was in a bit of a rush. You have to have capital letter, full stop, commas, whatever, بس I rushed it شوي. The first two I didn't really rush as much, so we'll look at that. We'll look at the first two more, like more in detail as an example, okay, of how to write it. So describe why hydrogen bonds form between water molecules. Obviously, the slightly positive hydrogen bond, the slightly positive hydrogen bond, hydrogen atom links together with a slightly negative oxygen. Why did I write bond? Atom, I'm sorry guys. In its vicinity. Okay, with a full stop. Vicinity. Therefore creating a hydrogen bond. Forming is better. Okay, so that would be a good answer to this. They're only telling you how to describe how it happens. So that's you describing how it happens. Explain why the arrangement of water molecules is different in ice and water. So this is improper, but ice is less dense than water, meaning its atoms are farther away from each other, whereas in water, since it is denser than ice, they are packed more closely together. Okay, that's the difference of how the arrangement of water molecules is. Guys, here they're telling you to draw arrows, okay? So, and صراحة, I'm not really sure how to do this, okay? So if somebody can email Mr. Luay and ask, that would be good. But, because I don't have his email. But what's it called? I'm not really sure about this because they didn't specify which is the hydrogen, which is the oxygen. It's very weird. So I'm gonna skip this one because I'm not sure, and I don't wanna yap about something that I don't know. Okay. Part D, the arrangement of water molecules in ice causes the ice to float. Explain how ice floating on the surface of a body of water affects the water in a way that is beneficial to the organisms in it. It's just like that question that always comes in the AMS about the river with the fish, the little picture of the fish and the ice on top and the insulation and blah blah. So ice floating on water acts as an insulator, keeping the cold out and the warm water under it, helping the animals living in it to maintain their body temperatures and living environments. Okay, now let's go back to number one. Number one, hello. Oh, okay. Okay, so describe the importance of the inner membrane, separating different regions of the mitochondrion and chloroplast. Oh, by the way, guys, on the last AP that I did, I had to type answers. Okay, but on some APs they give you the paper. You have to do it on a paper. It's like a written part. Okay, the multiple choice is all on computer now. Okay, from last year they started doing it on computer. It's through an application called Blue Book. To use Blue Book, you have to have either an Apple device, a Windows device. Yeah, just Apple or Windows. Okay. It can be even an iPad or a MacBook or a Windows laptop or something, anything, but it has to be Windows or Apple operating systems. Okay. And some exams you'll have to type, like AP Environmental Science, you have to type. But on AP Biology, you have to write because sometimes they tell you to like draw or do stuff like that, and it's hard to do it it's sometimes impossible to do it on a computer, which is why you do it on paper. But yeah. Let's start. This is more proper than the other one, so look at this more as an example of what you're supposed to do. So describe the importance of the inner membrane separating different regions of mitochondria and chloroplast. So the inner membranes of the mitochondria and chloroplast, the mitochondria, it's called cristae, and in the chloroplast, they are called thylakoids, separate different regions, creating distinct compartments for specific reactions. So each reaction has its specific compartment, a specific space where it works. This separation increases the efficiency of processes such as oxidative phosphorylation in mitochondria. So oxidative phosphorylation is just a name of a process that the mitochondria does, and the light-dependent reactions in the chloroplasts. Okay, you don't really have to state the name of the processes, but it's good. Okay, you can say that this separation increases the efficiency of processes in the mitochondria and chloroplast. That's fine. And folding of these membranes increases surface area. That's very important, allowing more enzymes and electron carriers to be embedded. So it allows more enzymes and the hydrogen ions to be embedded, and allows increase in ATP production. Okay. Based on the diagram, describe how the structure of these organelles controls movement across the membranes. Okay, so the inner mitochondrial membrane and thylakoid membrane contain protein complexes such as electron transport chains and ATP synthase. These selectively pump protons H+ across the membrane, creating a concentration gradient, because the membrane is otherwise impermeable to ions. The gradient is maintained and protons flow back through ATP synthase, driving ATP production. Okay, sorry guys, someone came in. I'm back now. So, based on the diagram, describe how the structure. Okay, so I already did that. Okay, so I'll go into detail about it. So, how these organelles, chloroplasts and the mitochondria, they control the movement across the membranes. Okay, so the inner mitochondrial membrane and the thylakoid membrane, okay, they contain protein complexes. Okay, they have this thing called the electron transport chain and ATP synthase. Okay, you don't really have to know. You can just say that the inner mitochondrial membrane and the thylakoid membranes. How can we make that simpler? The inner mitochondrial membranes and the thylakoid membranes in the chloroplast selectively pump protons, okay, so the H+, across the membrane, creating a concentration gradient. Okay, so it pumps the H+ from a place of high concentration to low concentration. Okay, because the membrane is otherwise impermeable to ions. So without these membranes, the folding membranes, the inner membrane of the mitochondria and the thylakoid membrane of the chloroplast, it's impermeable to ions, which is why it's very important. The gradient is maintained, so it's from high concentration to low concentration, and protons flow back through ATP synthase, driving ATP production. I missed that. Okay. Here, guys, basically, I didn't solve this because, yeah, I don't know. First, you find the volume 4/3 pi r cubed. Okay, for surface area 4/3 pi r squared. Okay, they already gave you the radiuses. You can keep them in millimeters. That's fine. Okay, just don't forget the units. It's millimeter cubed. Okay, and wait, I'm not sure what I'm not sure. I'm actually not sure which unit you should use for surface area. Wait, give me 1 second. Okay, guys, I have asked our dear friend ChatGPT, and it is millimeter squared. I was expecting it to be millimeter squared, but I didn't want to say something that was wrong, so I double checked it. Okay, and then here for the last one, after you've done the 4 pi r squared and the 4/3 pi r cubed to get the surface area and the volume, you just divide them to get the ratios. And yeah, I'm not actually going to do them because that will take too much time. The video will be long and nobody will open it in the first place. So yeah. So explain how the folding of the mitochondrial inner membrane and many individual chloroplast thylakoids are beneficial to a cell that is carrying out chemical reactions. Okay, so this is really very similar to the first one. Okay, I tried to make my answer a little bit different. So I said that the folding of the inner mitochondrial membranes into cristae, because the inner mitochondrial membranes, when they fold, they're called cristae, and the stacking of thylakoid membranes into grana. So when the thylakoid membranes stack like pancakes, it gives you grana, okay? They greatly increase the surface area, so that's like the main point. This allows more electron transport chains and ATP synthase molecules to be embedded, maximizing ATP production. You can really do without that sentence, and that's just extra yap for the College Board, but really, you don't really need all of that. You can just say that the folding also separates processes into distinct regions, and both of those reasons increase the efficiency of cellular respiration and photosynthesis, respectively. وبس, يعني that's all you have to do. That little sentence, this allows more electron transport chains and ATP synthase molecules to be embedded, maximizing ATP production. If you don't understand that, if you don't know what that is, that's fine. Just don't talk about it. You're fine as long as you put the first the first sentence and the last sentence. You're okay. Number 2. Yeah. So identify the process used to form the covalent peptide bonds. It's a dehydration reaction. I was too lazy to write a huge, big sentence. You can write the process that is used to form covalent peptide bonds that join amino acids together into polypeptides is called the dehydration or condensation reaction, where a covalent peptide bond is formed by secreting an enzyme and having it attached to both amino acids, one removing the hydroxyl group and one releasing a hydrogen atom, that then bind together to form water. Therefore, we release water to then join the two amino acids into one polypeptide chain. I just was too lazy to write that. The change in the amino acid sequence illustrated in figure 1 caused a change in the shape of receptor X. I wonder, I changed the R group. What is going to happen? Based on the R groups of the original and substituted amino acids, explained why receptor X changed its shape. The three-dimensional shape of a protein, particularly the tertiary structure, is determined by the interactions between the R groups. If you change the R groups, it will have an effect on those interactions. And since those interactions are the thing that's responsible for the shape will change. Okay? Using the template below that represents the 4 levels of protein structure, place an X on each level that is expected to be altered as a result of the amino acid substitutions. صراحة guys, I'm not very sure about this question. I think that it's this, that, and that. I'm not sure about the quaternary structure because I don't know if this forms a quaternary structure. You know, it's 5 amino acid sections of receptor, blah, blah, but they don't say that it's just amino acids, you know. So it's not 2 polypeptide chains linked together. It's just 1 polypeptide chain. So it should stop at the tertiary structure. It shouldn't go on to quaternary. That's what I think. So that's what I put. So I think it's this, that, and that. I don't think you should put an X on quaternary structure because I don't think it will be altered, because I don't think there's quaternary structure from the beginning, يعني من الأصل. Okay, I'm not sure. If anybody knows, please tell me, contact me, please. Explain how the amino acid substitution shown in figure 1 is most likely to affect the function of receptor X. So, basically, listen, I'm not really sure, okay? Like I said that the mechanism of effect, it will change the tertiary structure, okay? Obviously, I'm sure of that, it will change the tertiary structure, I'm 100% sure. The consequence for changing the tertiary structure, so the binding site is made up of a precise arrangement of R groups, and then if you change the overall protein shape, the substitutions in the binding site, like if you change the binding site, the binding site's shape will be altered, it will be changed. So it won't be able to bind to the specific protein that it was supposed to bind to before. Okay? So I'm not sure. And then the conclusion, this change in the shape of the binding site will disrupt the necessary complementary fit between receptor X and the specific molecule it is meant to bind, just like I said. And the specific molecule will no longer be able to bind to receptor X. That's what I came up with. So the final result, if the molecule cannot bind, the cellular response cannot be triggered, meaning the function of receptor X will be lost or severely diminished. This is a classic example of how a change in primary structure leads to a change in three-dimensional structure, which in turn abolishes or changes function. Guys, if you're going to write it in a list form like I did, okay, if you're going to write it like this, make sure to include bullet points, especially in the actual AP, okay? It's nice to have this in bold. It's good to have this in bold, and when the college board sees things in bold and like commas and organization, they give you marks. Okay, it's easier for them to correct, so they're in a better mood, so they give you better marks. Okay, you can put this all in like a big fat paragraph. Okay, you can make this shorter, you can play around with it. This isn't the only correct answer, you know, but just be careful with it. Okay, also that's the end of the video. Guys, make sure to study hard, study this video. If you don't understand this video, it's like a big problem. This particular quiz is very important, not just for our school exams, for the most important exam is the AP exam, especially for those who want to study outside the country. The AP is very important. Make sure you know how to answer on the FRQ, because the FRQ is like 50% of your grade, so be careful with this. Okay, thank you so much, guys. Have a nice rest of your day, and I'll see you guys tomorrow. Inshallah khair, inshallah we all get hundreds, we're all happy after tomorrow's exam.